Bismillahirrahmanirrahim. Assalamu alaikum warahmatullahi wabarakatuh. Good morning, respected organizers, distinguished colleagues, researchers, and physicians. I am Dr. Shahidullah from the College of Medicine, Southwest University, PR, China, and Department of Biochemistry, Human Medical College, Abbottabad, KPPA, Pakistan. Today, it is my great honor to present at the first international conference which is hosted at Women Medical College, Abbottabad, KPK, Pakistan. My talk today is titled The Beauty of Emerging Technologies in the Battle Against Cancer. So you know that cancer remains one of the most dangerous diseases worldwide with high mortality despite advances in treatment. Our research explores how nanotechnology, metal-based complexes, gene editing system, and artificial intelligence can transform the cancer therapeutic landscape. So our purpose and aim is to highlight novel strategies from silver nanoparticle in gastric cancer to ruthenium complexes in breast cancer, supramolecular nanocarriers for CRISPR-Cas9 delivery, and AI-driven diagnostics that collectively represent a multidisciplinary four-in-one system for future oncology. So here you can see we had proposed the hypothetical mechanism in 2018. Not only we had published, but it opens doors for oncologists. With the help of this proposed hypothetical mechanism, we explored that how nanoparticle enter into the body, what's its mechanisms, silver nanoparticle, how much cost effective and eco-friendly. Later on, 2022 to 2025, we run another experiment means to design a new project with the collaboration of Renmin Hospital, Wuhan, PR, China, which is also known as the city of COVID-19. So, you know that 5-fluorouracil last 2 decades ago, many scientists, researchers, and physicians, they are using 5-fluorouracil against cancers. And here you can see on the right side picture, She et al. 2021 published some nanoparticulate drug delivery system for 5-fluorouracil. So there are many shortcomings of 5-fluorouracil. So our group decided that how to reduce these shortcomings. For this purpose, we have had synthesized with the different ways silver nanoparticle. After careful examination, their physiochemical characterization and other phenomena, we concluded that we need to use green nanoparticle and chemical nanoparticle against gastric cancer. So for this purpose, firstly, we had purchased the human salines HCG27 and MGC803 from American lab. You can see here in this picture, the left side representation for cytotoxic effect of silver nanoparticle in the form of chemically synthesized and biogenic synthesized, while in the right side, the 5-fluorouracil individually and with the combination of chemical synthesized and biogenically synthesized. The red arrows indicated in the right side the significance of 5-fluorouracil with the combination of chemically synthesized and green synthesized nanoparticle. So the result showed that when we used 5-fluorouracil with the combination of green nanoparticle, so it has more effective as compared to individual as well as chemically synthesized silver nanoparticle. So then we move for malignant behavior of gastric cancer cell under silver nanoparticle individual, 5-fluorouracil individual, and with combination. So similar result we have received that silver nanoparticle synthesized with the help of green method had great impact with combination of 5-fluorouracil as compared to individual. As the same case you can see here, the gastric cancer cell apoptosis under silver nanoparticle and 5-fluorouracil. And for intracellular uh, reactive oxygen species of gastric cancer cell under silver nanoparticle and 5-fluorouracil. So collectively, I would like to summarize the whole story that our results showed that enhanced apoptosis, reactive oxygen species generation, and altered box BCL2 and P53 expressions in gastric cell lines, especially in the combination with 5-fluorouracil, is significant as individual or with the combination of chemical synthesized silver nanoparticle. So here our group suggested that silver nanoparticle could be a promising player as a standard in chemotherapy. In part 2, here we have synthesized 12 ruthenium complexes, and these 12 ruthenium complexes we had used for a new anti-tumor strategy in triple negative breast cancer. So our group filed a Chinese patent from these ruthenium complexes with strong anti-cancer activity. You can see here, these are the 12 ruthenium complexes from C1 to C12. C1 indication of ruthenium complex 1, C2 ruthenium complex 2, and up to C12. C12 means ruthenium complex 12. So you know that stability is one of the most important and key factor which affect the biological activity. So for this purpose, first we need to check the stability of metal complexes, especially ruthenium complexes. With the help of this stability, then we move ahead. So you know that we have a 12 complexes. So 12 complexes is very difficult to check one by one. So with the help of stability and with the help of other type of activity, we need to choose the best one. So after careful examinations from complex number 1 to complex number 2, so our results showed that complex number 8, complex number 2, and complex number 11 exhibit substantial anti-cancer activity, and their stability is good. So we checked the anti-cancer effect of ruthenium complexes against 4 different cell lines, including MDA, MB231, 41, HeLa, and U at 7mg. These are the 4 different cell lines. Why we check? Because we need the best result. And at the end, we move only on that cancer cell, and we use those complexes which having the good results. So here you can see the cell growth inhibition effect of ruthenium complexes were evaluated using 4 different cell lines, and these cancer cell lines were cultured and treated with each complex for 48 hours. And we used the method CCK8 assay to measure cell viability. After careful examination, after results showed that complex number 8, complex number 11 exhibited significant high potency in inhibiting cell growth across all tested cancer cell lines compared to other counterparts in the series of complexes. So next, we need to check to assess the cytotoxicity of these 3 complexes: complex number 2, complex number 8, and complex number 11. So here you can see for the cell viability and drug toxicity were assessed using a comprehensive live and dead assay employing fluorescent dyes. Our experimental finding revealed a significant increase in the number of decreased cells following administration of anti-cancer drugs, complex number 2, complex number 8, and complex number 11 compared to the control group. So you can see the control group is in the beginning. And among the 3 complexes, complex number 18 on top here, the red spots. The red spots show the cytotoxicity. So next, we check the cancer cell migration induced by ruthenium complexes that I have already told you that we have the 3 best complexes, which is complex number 2, complex number 8, and complex number 11. So we checked the inhibiting cancer cell migration, which is closely associated with blocking cancer advancement. In this study, here the impact of most potent complex on cancer cell migration using the own healing assay against these 4 cancer cell lines. So here we concluded again that complex number 8 emerges a particularly noteworthy showing robust inhibition of migration across all 4 cell lines. So you know that mitochondria is play a crucial role in energy metabolism and ATP synthesis. So for this purpose, we check the mitochondrial disinfection and ionogenic potential. So mitochondria, you know that it play a crucial role in energy metabolism and ATP synthesis, making them a key target in the development of chemotherapeutic resistance. So cancer cells often adapt to drugs by enhancing mitochondrial functions. So a major aspect of mitochondrial dysfunction is its involvement in apoptosis and cell death. So again, to verify if complex number 8 induced apoptosis was mediated through mitochondrial damage. So for in vivo, we have also examined the significant of tumor, significant of tumor growth and increase in survival rate of tumor bearing mice. So here we investigated the efficacy of novel drug treatment regime initiated on day 7 following the detection of a tumor in mice. This regime encompassed the administration of PBS as a control, cisplatin as a commercially available drug, and complex as a potent ruthenium complex is studied at interval of every 3 days for a total of 6 doses. So no alteration in the food or water consumption pattern of treated animal were observed. So the live mouse tumor growth and progression were monitored after the administration of control and treated group. So again, I would like to say proudly and interestingly that complex number 8 showed remarkable ability to prevent tumor migration to distant body site, surpassing the outcome observed with the PBS control. So pathology expression analysis of tumors and bearing cells, and I would like to skip this slide. So finally to check the side effect of this complex. Basically, we also explored that complex number 8 impacts on various physiological parameters in tumor-bearing mice. So the findings revealed that significant reduction in spleen size and weight among mice treated with complex number 8 compared to the cisplatin or PBS control group. This reduction suggested a potential modulation of the inflammatory response by the compound, possibly through a mechanism involving tumor growth inhibition and suppression of metastasis to the spleen. So we further investigated the pathological response of heart, liver, spleen, and kidney following complex AT administration. So the result showed that complex AT treatment didn't induce pathological changes in the heart, liver, or kidney. So in contrast, we can say that cisplatin treatment caused significant inflammatory cell infiltration in the spleen compared to cisplatin, and complex treatment didn't exhibit significant inflammatory cell filtration and was compared to PBS treatment. So in part number 3, right now I have very little time. I am going to very quick. Here, nanoparticle and CRISPR-Cas9, basically right now we have designed a new project. You know that CRISPR-Cas9 is also known as the Nobel Prize winner Jennifer Doudna 2022 techniques. So with the help of this, we can also invert it, and we can modify it, and maybe with the help of a gene editing tool, we can to cure different metabolic disorders. And you know that CRISPR hold immense promise, but self-delivery is a bottleneck. So we designed a supramolecular nanoparticle system combining with others like PEG and cyclodextrin polymers. So this system enhances biocompatibility, loading capacity, and nuclear gene release, making CRISPR delivery feasible in oncology. Here you can see in 2024, we have also proposed one mechanism, the hypothetical mechanism for CRISPR-Cas9, and Alhamdulillah, right now it is going, and we have received a very good result and the same result as we have proposed. In part number 4, this is our another group. They are working on artificial intelligence and bioinformatics. So here we can conclude that you know that artificial intelligence basically helps to integrate multi-omic data for novel drug targets. And application include early diagnosis in breast cancer, gastric endoscopy, and pathology workflow. And here you can see that we have very recently identified a new artificial intelligence breast cancer uh, like 5 years before it was developed. So at the end, I would like to say that together artificial intelligence enhances the precision oncology and speed up drug development. In conclusion, you know that these emerging technologies, nanomedicine, transition metal complexes, CRISPR, gene editing, and artificial intelligence are not isolated tools, but interconnected strategies. Together, they form a transdisciplinary approach in our ongoing battle against cancer. So this slide is basically we have collaboration with different countries, and we have published our papers in different journals like Nature, Science, Press, Nano, and so on. And this is about the speaker means me. To that, how many papers I have published, books and chapters, the impact factor, citation, the H index and I-10 index, and below is some award that I have received in the area of scientific field. So finally, I would like to conclude my presentation with a great acknowledgement and thank you for your attention. I also extend my sincere gratitude to the organizers of Women Medical College, Batabad, KPK, Pakistan, to provide me this nice opportunity and for any kind of collaborations or cooperation. I am always open and welcome. Thank you very much for your kind attention. Assalamu alaikum wa rahmatullahi wa barakatuh.